Hi, I'm Dr. Peter Crapo, and with the Fox Center for Vision Restoration, and I'm here with Dr. Jeff Goldberg. He's with the Bascom Palmer Eye Institute and the Interdisciplinary Stem Cell Institute at the University of Miami. So Jeff, first to start off, could you tell us a little about your background and what motivates you to work in the field of regenerative ophthalmology?
Sure. Well, first, Peter, thank you very much for having me. I started at Stanford, starting my MD PhD program in 1995.
Okay.
And it was right at the time that Christopher Reeve actually had his horse riding incident where he was spinally injured with a
Right.
very high spinal cord injury. And although I knew I was interested in neuroscience in general, that motivated me to start studying regeneration. I joined a lab there, Ben Barris's lab at Stanford, where we were using retinal ganglion cells, neurons in the eye, which project through the optic nerve, as a model system for regenerative failure, because the optic nerve doesn't regenerate just like the spinal cord doesn't regenerate. And through that experience, I also came to appreciate that there are so many ophthalmic diseases where regenerative failure really limits the healing of people's vision. And that's really what got me into regenerative ophthalmology, was sort of starting in thinking about regeneration in the nervous system in general, and then focusing my energy and interest in ophthalmology.
Yeah, there's a lot of parallels there. The spinal cord doesn't heal itself either, and the optic nerve, with the parallels with that, it sounds like you saw the parallels, but then working specifically with retinal ganglion cells that developed your interest in ophthalmology specifically.
Exactly right, exactly. And when it came time to decide what field to continue my career into after graduate school, I sampled different areas, of course, in medicine, and really realized that I enjoyed the particular opportunities and challenges in ophthalmology, and so it was really a perfect match.
Excellent. Well, maybe you can even go back a little bit further and tell us a little bit about some of your earlier research and then your current research projects and how those relate to what you did earlier on in your career.
Sure, absolutely. So, starting in graduate school, I began studying retinal ganglion cell axon growth and regeneration. I was very interested in both the extrinsic signals that promote regeneration, guide regeneration, as well as the intrinsic signals in the neurons themselves, in the ganglion cells themselves, that may regulate their ability to regenerate. And those two questions really served as the core questions for my graduate work at Stanford. Coming to Miami, I continued that, and that continues to be, I would say, one of the core sets of projects in the lab, is really trying to understand what regulates retinal ganglion cell regeneration and what can we do to enhance the regeneration in the adult to improve optic nerve repair. But we've branched out from that, and we now have a more comprehensive set of experiments of directions in the laboratory, all still revolving around retinal ganglion cell and retinal and optic nerve biology. We've begun studying in greater detail some of the signaling pathways within the neurons, and we've been using magnetic nanoparticles and taking a nanotechnology type approach to studying axon growth and signaling, what are called signaling endosomes inside the cells.
Little bodies inside the cells that carry signals, essentially.
Exactly,
Right.
exactly. So we're asking what role localization plays by manipulating localization using nanoparticles and magnetic fields.
Right.
We've begun to study retinal ganglion cell generation or differentiation from stem cells, or what are called retinal progenitor cells, precursor cells in the retina. And we're studying those both in the embryo, when retinal ganglion cells are normally found, as well as in the adult. And that's been really fun as part of the Bascom Palmer Eye Institute research group, but also now as part of University of Miami's Interdisciplinary Stem Cell Institute, as you mentioned. So we're studying how retinal ganglion cells form from stem cells, what controls that and regulates that, and then looking at transplantation of progenitor cells, retinal ganglion cells, other retinal cell replacement therapies.
Right.
And then the last component has been taking that up to the tissue level, tissue engineering level, where we're using three-dimensional scaffolds to try to ask questions about how retinal ganglion cells normally interact with their retinal neighbors, for example, amacrine cells.
Right. So it sounds like you've got a variety of projects that sort of run the gamut from the molecular level with specific, I guess you could call them cell signaling molecules within the cell or outside of the cell that they may then pick up, all the way up through the cellular level up to the tissue level where, and there's, I think, a great need at all of these levels. We need to understand what the signals are so that, and you're working on that, so that we understand how to tell the cells what to do in order to regenerate, all the way up to recreating tissue if necessary for instances where it may be necessary to fully replace. It sounds like you've got a full spectrum there.
Keeps us busy, but it's very stimulating. And I think that we derive a lot of synergy out of attacking the essential problem of the failure of repair in the visual system, attacking that from a few different directions where we can hope to gain some synergy through the different approaches.
Right. Great. So Jeff, as a follow-up question then, you talked about the intrinsic ability of the retinal ganglion cells to regenerate, to sprout new axons into the optic nerve and hopefully reconnect with the vision centers in the brain. And so what is that intrinsic ability? What is that built-in ability for us when you have a patient or someone who's had optic nerve damage? What is that inherent ability that they have? And then with the work that you've done, how have you been able to enhance that ability, and where do you think we need to go from where we're at in order to actually recapitulate function?
Right. I think that's a really important question. Normally, in the optic nerve, there's no regeneration, and so in diseases like glaucoma or optic nerve strokes or any other optic nerve degeneration, really the retinal ganglion cells have no intrinsic ability to regrow. And
virtually zero, then.
Zero, really zero. And as you mentioned before, it's the same thing as in the spinal cord, where once paralyzed, it's permanent. Well, in the visual system, in the optic nerve, whatever vision you lose from axon damage, it's permanent, because normally there's zero regenerative capacity. So that's a really important question. Normally there's no regeneration in the optic nerve, so any damage done to retinal ganglion cell axons, whether it's from acute injuries like optic nerve strokes or degenerative diseases including glaucoma, there's normally no regeneration, and that's certainly true both in the human disease, it appears, as well as certainly in the preclinical models of these diseases that have been studied. So there's been a lot of really important preclinical progress that's been made, and our work is only one piece of that. And in some of our recent studies looking at modulating the intrinsic regenerative capacity of retinal ganglion cells, we've been able to get a few % of fibers to regrow down the optic nerve.
Yes, that's certainly still an impressive difference from 0 to a few %, right?
Even to move from 0 to a few % is very encouraging.
Right.
And others in the field have made similar, or in some cases even dramatically more progress than that. There's some really beautiful work that's been done recently demonstrating, again, after optic nerve injuries in some of these preclinical models, that a significantly larger % of axons can be induced to regrow down the optic nerve and even get themselves most of the way back to the brain. So for the first time now, the field has taken a big step forward where we're not able to ask questions just about how to get the axons to regrow, but we're going to be able to start asking questions about how to guide them to the right spot in the brain, and how to get them to reconnect and reform synapses, their connections, with their natural targets in the brain. So we're just now coming up to the next really exciting step in this research.
Right, right. Yeah, so it sounds like there's sort of a series of steps that need to occur here, and sort of the first step is to get the axons to regrow, and we're maybe at the point where we can start worrying about further down the road, which is very exciting. I agree.
Very exciting.
Yeah.
That's right.
Excellent.
Very good.
Well, Jeff, thanks for joining us. As a final question, I'd like to ask you, as someone who's done seminal research in this field, where do you see the field going, and what are the research projects going on right now, the research that's happening right now, that you see as the most exciting, most promising, and where do you see all of this going in the field of regenerative ophthalmology in maybe 5 or 10 years down the road?
I think we're at a really exciting phase in research and in the development of this research towards human use. And I think there's been a real appreciation, even increased over these last few years, of our ability as scientists and scientists and clinicians to, in terms of areas, I think there's a lot of promising work looking at stem cells and cell replacement therapies, tissue engineering type therapies where we can help rebuild the retina and optic nerve. I think there's a lot of really beautiful work being done on drug delivery and the use of nanotechnologies to deliver drugs, to deliver new therapies. But as I mentioned, to some degree, some of the most exciting work has been watching the early stages of translation from what's being done in the laboratory towards human use. And I think over the next 5 to 10 years, what we're really going to see is a proliferation of early stage human trials where we really start to ask, hey, can we start helping our patients with all these advances?
Right. How effective are these potential therapies, and maybe we're at the point, you think, where in the next decade or so we'll actually be able to see some of the initial forays into how to actually treat patients then.
I think that's right.
Yeah, excellent. Well, Jeff, thanks for joining us.
Pleasure.
We appreciate it. So
It's a pleasure. Thank you.
It's good to have you.